Good morning. Welcome to the second session of the same chapter, that is molecular basis of inheritance. In the previous session we studied DNA, what is meant by DNA, and the DNA is composed of three components and how these three components are arranged to form an elongated chain that is said to be polynucleotide chain. And then we studied Watson and Crick model and the function of the DNA and central dogma. Now coming to the next is the packing of DNA. Now the DNA, human DNA, consists of 6.6 into 10 to the power of 9 base pairs. Then how to calculate the length of the DNA? Length of the human DNA, that is, this is the base pair 6.6 into 10 to the power of 9 base pairs into distance between the two nucleotides, that is 0.34 nanometer into 10 to the power of minus 9. So totally it comes 2.2 meters. This is the length of the human DNA. In case of Escherichia coli, 4.6 into 10 to the power of 6 base pair multiplied by the same thing. The distance between the two nucleotides is same 0.34 nanometer into 10 to the power of minus 9, approximately comes 1.52 meters. Here the length of the human DNA is around 2.2 meters, but the dimension of the nucleus is only 10 micron meter. So the length is greater than the dimension of the nucleus. So here you are going to understand how this DNA that is tightly packed inside the nucleus, because the size, the dimension of the nucleus is very small and the length is very long. We will see the package of DNA in prokaryotes. Here, in case of prokaryotes, the DNA is said to be double-stranded structure and it is circular structure, coiled and coiled again and again to form super coiled, and it is said to be naked because there is an absence of nuclear membrane. The four important aspects: double-stranded, it is circular DNA, highly coiled, therefore it is said to be super coiled, and it is said to be naked. So here what happens? DNA coils mainly due to the presence of polyamines. As a result of this, imagine this is the bacterial cell. As a result of this polyamines, there will be a continuous coiling and coiling so that it occupies or it confined to the very small space in the cell. Understood? So this is said to be, what do you call? Nucleoid. Nucleoid. This is said to be nucleoid, also known as genophore. This is said to be nucleoid or genophore. So the packing of DNA in prokaryotes, DNA is supercoiled mainly due to the presence of polyamines, and it is double-stranded, circular, naked, supercoiled. As a result of this, it coils itself to form it occupies a very small space inside the cell, that is said to be nucleoid or genophore. Now coming to the next is packing of DNA in eukaryotes. Here, in case of eukaryotes, there is a presence of 4 important components. One is DNA, second is histone, third is non-histone, last is very little RNA. Out of these 4, what happens? These 2 are the major components in eukaryotes. But in case of prokaryotes, there is a presence of only the genetic material along with non-histone proteins or nothing but polyamines, and these 2 assemble together to form a structure that is said to be nucleoid. But in this case, there is a presence of 4 components. Out of these 4, these 2 are the major one. So, what is meant by histone? Histone is nothing but it is a nucleoproteins. That means proteins associated within the nucleus. Therefore, it is said to be nucleoproteins. And these histones are very rich in lysine and arginine. There are 2 important amino acids, lysine and arginine, are very rich in histones. So, what are histones? It is a nucleoprotein very rich in an amino acid known as lysine and arginine. Now, these histones are of 5 types: H1, H2A, H2B, H3 and H4. How many types of histones are there? There are 5 types of histone: H1, H2A, H2B, H3, H4. Out of these 5, these 4 occurs in pairs. That means in double, 2 molecules of this, 2 molecules of this, like that, 2 molecules of this and 2 molecules of this, to form a structure. This is said to be histone octamer, also known as new body, NU, new body. So, what is meant by histone octamer? H2A, H2B, H3 and H4 assemble together in pairs to form a structure that is said to be histone octamer. Now, coming to the next part here, around 200 base pairs of DNA, that is wrapped like this, 200 base pairs of DNA wrapped around the histone to form a structure, and this structure is said to be nucleosome. So, what is meant by nucleosome? Nucleosome is made up of 2 components. The one is the 4 types of histone occurs in pairs to form histone octamer or new body. And imagine this is the new body, so that the DNA made up of 200 base pairs, what happens? It is completely wrapped over this. As a result of this, there will be a formation of a structure. This is said to be nucleosome. So, here what happens? The nucleosome is like this. It looks like beaded structure. So, here what happens? This is nothing but the DNA wrapped like this, and it extends also like this. So, this part is said to be nucleosome, And between the nucleosome, you can see there is a presence of DNA that also extends, and this is said to be what do you call linker DNA. Linker DNA is also composed of H1 histone. So now you have to understand this linker DNA and H1 protein, only this part. This is not a part of the nucleosome. It is only the supporting structure. Understood? So this is about the nucleosome. So what is meant by nucleosome? There are four important histones, H2A, H2B, H3 and H4 occurs in pairs to form a structure that is said to be histone octamer. Octa refers to 8 and it is also said to be new body, and then around 200 base pairs of DNA that is wrapped over this to form a structure that is said to be nucleosome. And then the nucleosome now looks like a beaded structures like this. ಹೇಗೆ ಒಂದು ದಾರದಲ್ಲಿ ಮಣಿಗಳನ್ನ ಪೋಣಿಸ್ತೀವಿ ಆ ಥರ ಕಾಣಿಸುತ್ತೆ. That is the beaded structures. So each bead is nothing but a nucleosome. So between the nucleosome you can see there is a presence of DNA that extends from here to here, that is said to be linker DNA. Approximately this linker DNA is made up of 80 base pairs and associated with the H1 histone. This is the structure of nucleosome, that is nothing but packing of DNA in eukaryotes. In eukaryotes what happens? There is a presence of DNA and histone. Histone is the type of a nucleoprotein, very rich in lysine and arginine, and here there are 5 types of histones. Out of these 5, these 4 occurs in pairs to form a structure that is said to be what do you call histone octamer or new body, and this new body is completely wrapped by this DNA and it is made up of 200 base pair to form a structure that is said to be nucleosome. And between this nucleosome there is a presence of linker DNA that is not a part of the nucleosome, so that the linker DNA, it gives only the supporting structure and it is associated with H1 protein. So now coming to the next part is how this coiling of DNA that increases the width. Now imagine here, this is the DNA strand. The width is how much? 20 angstrom or 2 nanometer. And here what happens? These two now results in the formation of this beaded structures. This is said to be what do you call nucleosome, so that the width increases to 10 nanometer because of coiling and wrapping. Again it starts here, it starts like this, coiling structures. This is said to be solenoid structure. Again the width is increases to from 10 nanometer to 30 nanometer. Again it coils, super coils like this. This is said to be super solenoid, and here the width is again increases because of super coiling. So it increases from 30 to 300 nanometer, and then again there will be a coiling takes place, and this results in the formation of chromatin. And here from 300 to 700 nanometer. So this is how the coiling of the DNA naturally increases the width of the chromosomes or the chromatids. So here what happens, this is the double stranded structure, this is the nucleosome, slightly increases, and then it starts lightly coiling structure, that is said to be solenoid structure. So from here 10 nanometer to it increases up to 30, and then again it coils, super coils, that results in formation of super solenoid, that results in 300 nanometer, and finally highly coiled structure, that is nothing but chromatids. So now the two chromatids, this is the single chromatid and this is one more chromatid, come close to each other during metaphase, and these two attach by means of centromere, and this structure is nothing but metaphase structure. This is only the temporary attachment. Temporary attachment, the two chromatids or come close to each other, there will be an attachment of these two chromatids that results in what? That results in with the help of centromere, and this stage is nothing but metaphase stage. So now the chromosomes are found inside the cell. When the chromosomes are found inside the cell, that is present inside the nucleus during cell division. When the cell is not dividing, what happens? This chromosome occurs in a form of a thread-like structure. That thread is said to be, what do you call? Chromatin material. Chromatin material. So when the cell is not dividing, what happens? The chromosomes occurs in the form of a thin thread-like or fiber-like structure, that is said to be, what do you call? Chromatin. So there are, if this chromatin is stained, that means we have to add color, stain, by using aceto arsine or acetocaramine. When we add these two stain, what happens? There will be a formation of two types of chromatin. One is like this, loosely coiled, lightly stained. Second one is tightly coiled, darkly stained. How many types of chromatin materials are there? There are two types, loosely coiled, lightly stained, so that it absorbs very little amount of stain. This is said to be euchromatin, tightly coiled, and it absorbs large amount of stain. This is said to be heterochromatin. This is more active, so that it helps in transcription. This is inactive. So the differential staining technique, when we add this stain, some of the chromatin now becomes lightly stained, some of the chromatin becomes darkly stained. The differential staining technique is said to be heteropycnosis. What is meant by heteropycnosis? It is the difference in the staining technique is said to be heteropycnosis. So this is about the chromatin material. Chromatin is nothing but thin fiber-like or a thread-like structure that is present inside the nucleus. When we add stain by using these two types of stain, so one is said to be euchromatin, second one is said to be heterochromatin, and the differential or the differential staining technique, that is said to be, what do you call? Heteropycnosis. Now we will move on to the next concept, transformation. transformation experiment by Frederick Griffith. Griffith experiment. What is meant by this transformation? It is the movement of genetic material from one organism to another organism is said to be transformation. Movement of genetic material from one organism to another organism. It is very important. They will ask for 5 marks. So, what is meant by transformation? It is a movement of genetic material from one organism to another organism, and here Frederick Griffith identified this phenomena while he was working with a type of a bacteria that is said to be Streptococcus pneumoniae, which causes pneumonia. It is a type of a bacteria. So, this bacteria which causes pneumonia, and this occurs in 2 forms. One is said to be S strain, that is the smooth strain. Second one is a R strain, that is the rough strain. Smooth strain, rough strain. This smooth strain is said to be virulent. That means, imagine the bacteria is like this. Around the bacteria, there is a presence of a capsule, so that it is said to be pathogenic, so that it causes pneumonia. This R strain is said to be non-virulent because there is an absence of capsule. It does not cause any pneumonia. So, Griffith identified a phenomenon called transformation while he was working on a type of a bacteria that is said to be Streptococcus pneumoniae, which causes pneumonia, and this bacteria occurs in 2 forms, S strain or the smooth strain, R strain, rough strain. So, S is said to be virulent because there is a presence of capsule around the bacteria, and here the this is said to be non-virulent because there is an absence of capsule. So, now what he did is Griffith conducted experiments on mouse. What he did is live S strain is injected into the mouse. What happens? If the live S strain is injected into the mouse, what happens? If the S strain is injected, live S strain is injected into the mouse, it dies because it is a virulent character. Then heat killed S strain is injected into the mouse, it does not die. Then R strain injected into the mouse, it does not die. And the last one, S strain mixed with, or the heat killed S strain mixed with R strain and he injected into the mouse, it dies. What happens here? He did 4 different types of experiment. What he did is, he took live S strain injected into the mouse, it dies because S is a virulent. Heat killed S strain is injected into the mouse, it does not die, heat killed. And then next is the R strain is directly injected into the mouse, it does not die because it is non-virulent. Here what happens, heat killed S strain is mixed with the R strain injected into the mouse, it dies. Here what happens, heat killed S strain, so that here there is a presence of genetic material, that is the DNA. There is a presence of DNA. Now, genetic material enters into the R strain and changes this R into S strain. As a result, it causes pneumonia, finally results in death. Later, Avery et al. Continued this Griffith experiment, and what they did is they prepare an extract which contains DNA, RNA and proteins from S strain mixed with the R strain. After few hours, there will be a transformation takes place. This is the first experiment. What they did is they prepare an extract which contains DNA, RNA, proteins from the S strain, and then this is mixed with the R strain. And there will be a transformation takes place. In the second extract, what they did is same thing, DNA, RNA plus proteins that is present in the extract. This is mixed with RNase and protease enzyme. Same thing mixed with the R strain. What happens? There is a transformation takes place. There is a transformation takes place. So that this RNase destroys the RNA enzyme, protease destroys the protein, but still there is a presence of DNA, so that the DNA from the S strain enters into the R strain, changes the R strain into S, that results in transformation. And the last extract, DNA, RNA, proteins mixed with DNase enzyme treated and again mixed with an R strain. Here what happens? There is no transformation. So, no transformation. That means here what happens? DNase enzyme that destroys the DNA. So, in the absence of genetic material, there is no transformation, which proves that the DNA acts as a genetic material. So, this is about the Avery et al. Experiment. So, what is meant by transformation? It is the movement of genetic material from one organism to another organism. First, the Griffith conducted experiments on mouse. So, what he did was, here live S strain is injected into the mouse. It dies because it is virulent, and then the heat killed S strain is injected into the mouse. It does not die because it is heat killed. If the R strain is injected into the mouse, it does not die because it is non-virulent. If the heat killed S strain that is mixed with the R strain, then there will be a transformation takes place, because what happens, the genetic material from the S strain enters into the R strain, changes the R into S, so that it becomes virulent. But he was unable to give some proof for the biochemical components. Later, Avery et al. Means is French. Avery et al. Conducted or continue this experiment, and they prepare an extract that is from the S strain, that is which contains DNA, RNA and protein, and he divided into 3 groups. In the first group, what happens, it is mixed with the R strain, there is a transformation takes place because there is a presence of DNA. In the second part, what they did is they mixed with the extraction is mixed with the RNase and protease, so that this RNase destroys RNA, protease destroys protein, but still there is a presence of genetic material, so there will be a transformation takes place. In the last group, he added DNase enzyme so that it destroys the DNA. So there is no transformation. That indicates in the absence of genetic material, there is no transformation at all. So in the absence of genetic material, there is no transformation, which proves that DNA acts as a genetic material. So in this session, I hope you understood how this DNA that is packed inside the nucleus, that is in both prokaryotes and eukaryotes. In case of prokaryotes, there is an absence of nuclear membrane, so that it is confined to a very small region inside the cell, that is the bacterial cell, that is said to be genophore. And in case of eukaryotes, there is a presence of nucleosome model, that is nothing but histone octamer, so that this histone octamer is said to be new body and it is completely wrapped by DNA, and between this nucleosome there is a presence of what? Linker DNA. And how this chromatin material, that is stain, there are 2 types of chromatin material, heterochromatin, euchromatin. And then coming to the last is the transformation experiment. It is the movement of genetic material from one organism to another organism, Griffith experiment, and then Avery et al. Continue the Griffith experiment and they proved that DNA acts as a genetic material. Thank you.